Alright guys, so this is another video where we're going to talk about biomolecules. Just a quick review. Again, all these videos are very quick reviews over the units that we've covered so far. Make sure you look at your notes and other resources to get more information if you think you might need it. Okay, so carbohydrates. We said carbohydrates were just organic molecules, okay, that include sugars, starches, and cellulose. We said starches are long carbohydrates. Cellulose is involved in the formation of plant cell walls. Sugars, especially glucose, is what's made during photosynthesis, and it's what we use to make ATP. Remember that. Carbohydrates are also classified in depending on how many molecules are in the carbohydrates. We said that the monomers, so the small units that make up the big polymer, are called monosaccharides because they're only made up of one sugar, and we can see here. Okay, we call this a ring. So carbohydrates their shape is actually a ring. Okay, and then they're made up of carbon, hydrogen, and oxygen, and the ratio would be 1 to 2 to 1, right? So if there's 6 carbons, there's going to be 12 hydrogens, and there's also going to be 6 oxygens because of the ratio that we just mentioned. Okay, so again, they're made up of carbon, hydrogen, and oxygen, and the monomers or single units are called monosaccharides. Okay, and when you put a bunch of monosaccharides together, or at least 2 in this case, you create a disaccharide, which is 2 sugars. Okay, and then if you put more than 3 or more together, then you're going to create what's called a polysaccharide or starch in this case. Okay, polysaccharide, poly meaning many, saccharides meaning sugars, so many sugars. Okay, so again, carbohydrates are just sugars. Okay, and the monomers are monosaccharides. When you put 2 monosaccharides together, you call them disaccharides. When you have 3 or more, you call them polysaccharides. The function of a carbohydrate is going to be to provide fast energy for the body. It also stores energy, but very, very, very small amounts. Okay, and supports a lot of the structures in our body, or in other organisms like plant cell walls. Okay, like it says right there, and also it's in the cell membrane as markers, so it's used for signaling as well. Okay, lipids are just fats and phospholipids. Okay, fats, oils, wax. Okay, phospholipids, we know that's part of the cell membrane. Okay, but lipids are just fats. That's the short name for lipids. That's what you need to remember. You also need to remember that the phospholipid is made up of this triglycerides, right? And triglycerides, all it means is that you have one central molecule which is called glycerol. Okay, glycerol, which is the monomer. Okay, and then you have 3 molecules coming out of there, right, 3, and those are called fatty acids. Okay, so fatty acids. So again, lipids are fats. They're made up of glycerol and fatty acids. Okay, that's the monomers of lipids. They are made up of carbon, hydrogen, and oxygen, but they don't go within the same ratio as carbohydrates. Okay. Another thing is that lipids, their function is to store energy. That's their primary function. They will store energy. They will insulate our bodies, and they will also let me close these out. They will also function as hormones, and they are a very, very, very important part of our cell membrane, which controls what goes in and out of our cells. Alright, so that's carbohydrates, that's lipids. Remember, they're both made out of carbon, hydrogen, and oxygen, or CHOs. It's just that carbohydrates have a ratio, and they are made up of rings. Okay, lipids are fats, and they are made up of glycerol and fatty acids. Triglycerides, 3 fatty acids. Diglycerides, only 2 fatty acids. Proteins are just molecules that provide structure, and they allow us to they allow all the chemical reactions to happen inside of our bodies. We talked about proteins being enzymes and helping us in speeding up all the reactions that we need. Okay, their monomers, or the small units that make proteins, are amino acids, and you should We're very familiar with that, especially after going over protein synthesis. We said that amino acids are put together in the ribosomes to make polypeptides, which are proteins, right? So amino acids are the monomers, okay, the small units that make proteins, and the polymers we call polypeptides. Okay, remember, a lot of amino acids create a polypeptide, and that's what proteins are. Their function, like I said, there's a lot of functions for proteins. Enzymes, they are part of the cell membrane, part of the cell. They are very important in cell signaling for our immune responses. I mean, proteins, without proteins, you wouldn't be able to grow hair, you wouldn't be able to grow your nails, everything. There's tons and tons of functions for proteins. But what we need to remember is that their monomers are nucleic acids and their polymers are polypeptides. They are made in the ribosome, and they are very, very important in all the chemical reactions that happen inside of our body, especially because of how enzymes work. We said enzymes are proteins, so enzymes are able to speed up these reactions. Without enzymes, we wouldn't be able to digest our food or stay alive. Okay, and we'll go into enzymes a little bit, into more detail in a little bit. Okay, nucleic acids, we talked about too, DNA and RNA. We talked about them and how important they are when it comes to making proteins, right? Again, the picture here is of DNA molecule. The structure is a double helix. And you should be able to identify all the components that are labeled there, nitrogen bases. The G always bonds with a C, the A always bonds with a T, unless you're talking about RNA, and then T's are replaced with U's in there, uracil, right? The backbone is made up of sugar and phosphate. The bases are attached through a hydrogen bond, but nucleic acids, again, they're just DNA and RNA, and they're responsible for carrying or storing our genetic information, okay? Their function, well, let me come back to this picture. Okay, the monomers, remember that they are made up of nucleotides. So if we were to just grab this little chunk here, that's called a nucleotide. And a nucleotide has a phosphate, has a sugar, and it has a nitrogen base. And when you put, so phosphate, sugar, nitrogen base. Okay, and when you put a bunch of these together, then you're gonna create a polynucleotide or a nucleic acid. Okay, the monomers of nucleic acids are called nucleotides. The polymers are polynucleotides. And again, when you're reviewing biomolecules, make sure you look at your graphic organizer. If not, create one so that you can organize the monomers for every biomolecule, the polymers for every molecule, the functions and the structure of every molecule. Okay, there's only 4: carbohydrates, lipids, proteins, and nucleic acids. They are all very important, and we need them in order to stay alive, and for the organism or other organisms to in order for them to stay alive and make everything that they need in their bodies. Okay, nucleic acids, their function again is to store and retrieve genetic information, also to transport it, because we learned about that in protein synthesis and how RNA actually transports the genetic information out of the nucleus, because DNA is too big to get out of the nucleus and it can't go out. If it goes out, remember, it denatures, right? All right, so let's talk a little more about enzymes. Enzymes are actually proteins. We just mentioned that a couple of minutes ago, right? And what they do is they speed up reactions, okay? And they do that by lowering the activation energy. And you can see here, right? You can see it here. Without enzyme, then you follow the red line. It's like a roller coaster. You got to go way up there, and then you can fall down. Yes, and that takes a long time to go up there. But with an enzyme, we can reduce the activation energy, so the hill is actually smaller. And you get the same result, it's just that you get it much faster. Okay? And that's what enzymes do. Enzymes lower the activation energy for these chemical reactions to happen. Okay? Remember that they are very specific. So we talked about enzymes having an active site, okay, and a substrate or whatever goes in to the reaction will attach to the enzyme, okay, at that active site, okay. Then the enzyme will either break it apart or put it together, and then it will release it. Okay, the product will change, the substrate changes, but the enzyme never changes. If this enzyme changes, then it can denature. Okay, the enzyme will keep going and going through the reactions until it actually denatures or destroys itself or gets destroyed. Okay, the only thing that changes is the substrates or the things that go into the reaction. Okay, and then what comes out of the reaction we call products. Okay. So things that affect enzyme activity are pH, temperature, enzyme, and substrate concentration. We talked about pH being a substance being too acidic or too alkalic. We actually tested temperature by using our hands, and we learned that sometimes it speeds it up, sometimes it slows it down. And the enzyme and substrate concentration, we also did a lab over that. Depending on how many people were breaking the toothpicks, we could break more toothpicks. Depending on how many toothpicks we had in the lab, that was the amount that we were able to break. So again, there's things that affect the rate of the enzyme. Okay, pH, again, there's always going to be an optimal temperature, optimal pH. After that, you're going to start slowing it down until it actually denatures. Okay. All right, guys. So that's it. That's biomolecules. I hope it helped. Look at other resources. Look at your PowerPoints. Look at your notes. Make sure you review, and let me know if there's another video you guys want me to make for you guys to review.